Welcome to Biology My Passion. I am Soumya Harikrishna. We will learn Biomolecules chapter now. All living organisms, if you check, they are all made up of the same elements and compounds, whether it is plant or animals, multicellular organisms or unicellular organisms. So, we all have mainly carbon as a major component of all our molecules. So, these carbon containing compounds present in living tissues are collectively called biomolecules. But if you analyze the elements present on the earth crust, they are not different from our components. So look at this chart given in your textbook. The compositions are same, but only thing their amount may vary. For example, in living tissues, the amount of carbon and oxygen are very high compared to the amount available on the earth crust. Now, how do we find this composition? It is by elemental analysis. So analysis can be done in two ways. First way of analysis is of living tissue for organic compounds or organic molecules. If you want to analyze, how do we do that? You take a small piece of living tissue, maybe a plant tissue or an animal tissue or a part of a liver or whichever tissue you want to analyze. Then you use a mortar and pestle to grind it using something called a trichloroacetic acid. You know, last year in carbon compound chapter you learned acetic acid is CH3COOH. So one hydrogen removed, it becomes CH2Cl COOH. One more hydrogen, it will become CHCl2COOH. But if all the three hydrogen are replaced, then we get CCl3COOH. This is called a trichloroacetic acid. Why are we using trichloroacetic acid? It is an organic solvent. So many compounds can dissolve in this acid. So now after grinding we get a thick slurry. So that semi-solid substance is called a slurry. Now we have to filter it using a cheesecloth. So you take a test tube and you take one funnel. Inside that you put a filter paper or a cheesecloth and you put the slurry on it and you will get two fractions. One fraction you will get inside this test tube. This fraction is called a filtrate. Whereas what is staying down here, it's not coming down, it is staying here. This is called a retentate. Okay. So in this case, the filtrate has everything which is soluble in acid. So we can call it as acid soluble pool. Whereas this is called a acid insoluble pool. So whatever could dissolve in this trichloroacetic acid, they all have come down. That is called an acid soluble pool. We can see many small molecular weight substances in it. For example, they are called biomolecules. Micro means small. They are micro. That means their molecular weight is 80 Dalton. Dalton is the molecular weight formula to 800 Dalton. They all will be in the acid soluble pool. Whereas this retentate mainly consists of macromolecules. You know what is their molecular weight? It is more than 10,000 Dalton. Okay. Now, one thing here we have four main substances. One is polysaccharides, means the complex carbohydrates will be there. Apart from that, proteins are there. Then nucleic acids are there. Nucleic acids are the genetic materials, DNA or RNA, and lipids are also there. Okay, but here the lipid is slightly different from the rest. The rest are all purely macromolecules with a molecular weight more than 10,000 Dalton. But lipids have molecular weight less than 800 Dalton. So where it should come? It should come to the filtrate. But unfortunately, it could not come to the filtrate. So this is a question. Even though lipid is a micromolecule, why is it present in the acid insoluble pool? The reason is, you learned the Singer and Nicholson model of plasma membrane or cell membrane in the previous chapter. There we learned the membranes have a lipid bilayer, right? So lipid is not found as such, they are integrated as part of the membranes. So when we are grinding using this trichloroacetic acid, we are disrupting the cell membrane, and these molecules will come and join together to form big molecules. They will not be able to come down through the filter paper or the cheesecloth. That's why they stay here in the filtrate, retentate. So the lipids are low molecular weight, or they are biomolecule, but they are staying here. Now there is a second way of analyzing the tissues. Suppose you want to find out the inorganic substances present in it, or elemental composition. If you want to know, you have a different destructive process or analysis. Okay, here only a part of the tissue we took, ground, and we found out. But the second thing, whether it is a plant, usually when if you want to know the components of a plant, what you have to do is first to weigh the plant. That fresh weight is called a wet weight, because that contains the water also. Weight of the water keeps varying. So that is not an accurate value. So what we have to do, now we have to dry that living tissue in sun, and we allow all the water to evaporate. Then you weigh. The weight will come down to almost 1/7, because 70% is water. Okay, so in that case we get the dry weight. Dry weight is accurate because there is no fluctuation of water level in that. Now this dried one we have to burn into ash. So that's why I call it's a destructive process. So once it is completely burned, then you can analyze for different compounds and analysis you are doing in chemistry. So chemist point of view, the second method is done in order to find out the radicals or other elemental composition. But for biologists, basically we have to go for this, where we can distinguish between biomacromolecules and biomicromolecules. Let's see the first micromolecule, biomolecule, that is amino acid. So if suppose this is one amino acid, many amino acids are connected together to form a large molecule called a protein. So when you break down protein, you will get individual units called a amino acid. So what is the structure of amino acid? Amino acid is actually a methane substituted or substituted methane. You have learned the structure of methane last year in carbon compounds chapter. So here 4 hydrogen are satisfying 4 valencies of carbon. If you are going to substitute this hydrogen is substituted by an acid group, which is the functional group of acid, carboxylic acid, COOH. This hydrogen is substituted by NH2 group. NH2 group is amino group. So that's why this got the name amino acid. Now 2 more hydrogen are there. This hydrogen is substituted by R. R is a variable group. That means it will keep changing in different amino acids. And this H is a constant there. So these 3 are constant for an amino acid. This is the variable group. So since we have substituted the hydrogen in a methane, amino acid is called what? The substituted methane. And also both carboxylic acid group and amino group are on the same carbon. So it is the alpha carbon. So it is called the alpha amino acid. Now this is the structure. These 3 components never change. Only variable groups keep changing. Depending upon that, I am telling you 3 examples. First, the variable group is a hydrogen. If variable group is hydrogen, the rest 3 same. Amino group, carboxyl group, hydrogen. Here also hydrogen, then it is called a glycine. That is one amino acid. Then next case, these 3 are same again. Only the variable group has changed from hydrogen to CH3. CH3 is called a methyl group. So it is otherwise called then it is called a alanine. Then CH2OH is the R group, then the amino acid is called a serine. So depending upon this, 20 different types of amino acids can form. This R group will change, rest 3 will remain the same. So we are studying only 3 examples. If it is H, glycine, if it is CH3, alanine, and if it is CH2OH, it is serine. Another classification of amino acid is based on their nature, that is pH. So there are acidic amino acid. Acidic amino acid means in a normal one we know that neutral. So acidic, or we can say basic or neutral. In neutral, the COH and NH2 groups will be the same. So they balance and form neutral. For example, valine is an amino acid which is neutral. Okay. Or alanine, glycine. Coming to acidic. Acidic means 1 amino group is there but 2 carboxyl group are there. Monoamino dicarboxyl group. So acidic group is more, so amino acid becomes acidic. Example we can say glutamic acid and aspartic acid. Whereas basic amino acid will have more amino group and a less carboxyl group, that is diamino monocarboxyl group. For example, here lysine, arginine, all these are examples of basic amino acids. Okay, so you just have to learn the names and their examples. So acidic, glutamic acid, aspartic acid, basic lysine and arginine, neutral valine, alanine and glycine. Now another classification of amino acid, or the third type. First type was based on the R group, second type this, third type is the aromatic. They have R group as aromatic group, aromatic amino acid. A stands for amino acid. Example only you have to study. One is tyrosine, then tryptophan and phenylalanine. Just learn the names. Tyrosine, tryptophan and phenylalanine are the aromatic amino acids. One more concept we have to learn in amino acid is their ionic nature. It is ionizable in nature. For example, if this is one amino acid, HCOOH, okay, this is R, NH2. Okay, so this NH2 can acquire a proton and it can become NH3 plus. So this is one form of ionic form. Another one is NH2, but here it can become negative, COO minus, losing one proton. So that also can be negative. So both are ionized condition. Whereas these amino acids have a unique property that they can attain both positive and negative charges at the same time. This condition is called a Switter ionic form. That means, suppose this is 1 COOH NH2. Okay, from hydrogen means one proton, one plus and one minus, one electron. So from this one proton is going, so it will remain as minus. That same proton is taken by this amino acid, amino group, then it becomes NH3 plus. So whatever is going from here taken here. So at the same time it has got both negative and positive charges. This condition is called a Switter ionic form. Hope you understood the concepts about amino acid very well. If so, please like, share and subscribe to my channel Biology My Passion.